Good morning, good afternoon, good evening. We're glad that you could join us for today's HESI's Environmental Epidemiology Committee webinar series. Today's webinar is going to be focused on the current efforts by the American Petroleum Institute, CropLife International, and Health and Environmental Sciences efforts to increase the impact of epidemiology in risk assessment. Before we begin, just a brief disclaimer. A few logistics. This webinar is being recorded. It will be posted to the Environmental Epidemiology Committee's website. You can see the link there. Throughout the presentation, please enter your question in the question box. After each speaker, we'll have time for 1 or 2 clarifying questions. And then hopefully we'll have some time at the end of the webinar to have a little panel discussion and Q&A with all the speakers. If you have any questions about the HESI project or anything, feel free to contact Sandrine Deglin. Today's webinar will go in this order. We'll first hear from Dr. Sandrine Deglin, who will share about HESI's efforts. Then we'll hear from Omobola Mudasiru from API. And then we will end with Dana Sargent from Bayer, who will be presenting CropLife International's effort. And hopefully, again, we'll have time for a panel discussion and Q&A. And so without further ado, I'd like to introduce Dr. Sandrine Deglin, who has joined HESI in 2018 as a scientific program manager. Prior to joining HESI, she worked with the Environmental Public Health Program at the State of Alaska Division of Public Health for 4 years, where she managed the program for nearly 2 years. Prior to that time, Sandrine spent 7 years as a consultant in the toxicology and mechanistic biology group of Exponent, Inc. Sandrine is currently involved in 5 different HESI committees focused on human health and ecological risk assessment. Outside of work, you will likely find her whitewater kayaking in the US or abroad.
Thank you, Connie. Good morning, good afternoon, everyone. So I want to make sure we were having some issues with slides changing. I hope you guys could see the slides. I know they were not changing for me, but so thank you for joining us today. In the next 10 minutes or so, I would like to take you very quickly over what we've been doing over the past 2.5 years with HESI's Environmental Epidemiology Committee. So the committee was created at the end of 2017, and the action of the committee is to take action to help realize the full potential of epidemiology and exposure science in risk assessment and regulatory decision making. So it's a broad mission, and it sounds like a tall order, but when we started, we thought that we should probably try and figure out the environment and the landscape in which we were going to evolve. So I think we are all in agreement about the fact that epidemiology is a critical tool when it comes to evaluating environmental health risks. And if you look at the past 20 or 30 years, there's been multiple efforts to try and make epidemiology more impactful in the field of risk assessment and decision making. There's been frameworks published, papers, guidelines, and in spite of this, EPI data are still seldom used when it comes to quantitative risk assessment and the development of public health guidelines. Toxicology is usually the core of what is used for these guidelines. And the question is, why is that? So what are the barriers to that change, to making EPI more central to risk assessment? And how can we overcome these barriers? So with the committee, we decided to have an approach based on the organization of focus group meetings with different experts. So over the course of 2.5 years, we organized 5 different focus groups, 2 in 2018, 2 in 2019, 1 in 2020. And as you can see on the map, we had a nice representation of federal agencies and state agencies, as well as universities and different private companies. I will say that every single HESI project has to be tripartite with members of at least government, industry, and academia. So we did just that. And we did our last meeting in 2020 with funding agencies was a virtual meeting. So you can see it here in green with Autism Speak, March of Dimes, Michael J. Fox Foundation, and NIH and IEHS. So we had a good representation of sectors, and we also had a good representation in terms of expertise. So in the next few minutes, I want to focus on the 4 first focus groups that we organized with scientific experts. I'll touch on the funding agencies a little bit later. So in those 4 focus groups, we engaged experts such as epidemiologists, risk assessors, exposure scientists, toxicologists, and statisticians. And our goals were to engage them in a dialogue, in a conversation that was centered around a case study or a couple of case studies. And we want them to see how they were approaching the problem and how they were thinking. And one of the goals was to help them better understand each other and how they thought and how they worked. So we wanted epidemiologists to better understand risk assessors and vice versa. In the process, we wanted to figure out what the roadblocks to the better integration of epidemiology in risk assessment was or were, and what kind of incentives could be implemented to overcome these roadblocks. And of course, since we're interested in the long term, we also wanted to try thinking about a strategy to make a significant impact in this field. So each of these meetings was a day long, so there was a lot of conversation, lots of exchange, and many ideas were brought up. So here I'm just giving you sort of a distillate of what came out, and those 6 ideas or 6 challenges were those that came back over and over again along these focus groups. So the first one, or the first obstacle to integrating EPI to risk assessment, is the lack of transparency of research protocol. So, for example, if you're a researcher or you're a risk assessor and you read an EPI study, it's not always easy to figure out what has been done. Then there's the issue of publication bias, since there can be sort of an incentive to publish new exciting research over confirmatory research, for example, even if sometimes data gaps could be filled by publishing confirmatory research. Then there was the question of funding issues and the question of whether the best proposals always get funded, and the problem of data access and transparency. So if a researcher wants to reanalyze a data set, for example, well, access to this data is not always easy. And in a way, it's understandable when you think that some researchers built an entire career on one or a couple of data sets. And then the big one was the lack of communication across disciplines. So we realized that all of these disciplines I mentioned earlier are very siloed, and there's not a lot of dialogue among them, when if they were dialogue, they could leverage each other's work. And there is a will for it, or at least there appeared to be a will for more communication among these different expertise. And then the last one is insufficient cross-training. So we discovered that epidemiologists do not know much about risk assessment or what risk assessors need, and risk assessors may not always be able to interpret epidemiology study well or complex statistics. So when we took a closer look at these 6 challenges, 3 of them were identified as being within reach of funding agencies. So we thought, well, why don't we try to speak to funders? And so that's what we did. So in 2020, we organized one more focus groups with different funding agencies. And again, it was a day long, of quite a few hours long. It was a virtual meeting, and we had a pretty rich conversation. So here, I just want to talk about the communication aspect that was brought up. And there seemed to be a will to open communication channels between funding agencies and regulatory agencies. So if an agency like EPA is lacking data on a certain chemical, it would be nice to relay that to a funding agency, let's say NIH, so that they know there is a need there. Another point that was brought up was that research funded by advocacy groups depends on stakeholders' understanding of epidemiology. And so it's important to open another communication channel there to make sure that the research that will be undertook is oriented towards very defined gaps and needs. Finally, we discussed the fact that epidemiology is not as well understood as toxicology, and so some training could be useful to address that. And finally, that epidemiologists may not know where they fall short. So once again, some interventions could help, like workshops or different types of trainings. So in the end, whether with scientists, regulators, or journal editors or funders, everyone there's a common goal, which is to protect and promote the health of the public. And so with that in mind, and when we looked at the big picture of what we learned after these different focus groups, we came up with this diagram that's based on a theory of change analysis. And I'm not expecting you to read every single box, but to walk you through, at the top in red, you have our ultimate goal, which is increasing the impact of AP in risk assessment. Below that, the line of blue boxes is a list of assumptions that have to be fulfilled so that we can reach that goal. Below that, you have different outcomes or short-term goals that need to be met so that we can fulfill those assumptions. And the white boxes in the bottom half of the diagram is really the nitty-gritty of what has to be done. So different actions that have to be taken to reach those intermediate goals. And the red or colored circles are the different actors that are involved. So this emphasizes that we're not here to point the fingers at epidemiologists or tell them what to do, but in the end, if we want to better integrate AP to risk assessment, it's sort of a shared responsibility, and it's going to have to be a common effort among different actors like academia and industry and academic department heads, journal editors, funding agencies, and so forth. So as a committee, we have a few next steps, and those are very general, and we've been discussing, some more concrete actions. But first, we want to publish the lesson that we've learned over the course of these focus groups, and then try to take action to create and increase cross-sectoral and cross-expertise interactions. So we have some ideas that we're sort of cooking with the group. That it's too short right now to talk about it, but if you're interested, I can talk about it offline with you. And we're going to keep doing outreach to communicate the importance of epidemiology and the full place that epidemiology could take in risk assessment and decision making. So in the end, I think ultimately what we need to make stride in this field is we need a sea change. We need to think differently about this problem. And so if any of you have any idea on how to make a positive impact and a difference in this field, please share your ideas with us or even join our group. The door is open. And in the end, this is a multi-sectoral and multidisciplinary effort. And I don't think it's limited to scientists. We need people who are thinkers and problem solvers and who can think differently. So if you're interested, please let us know. And with that, I can take a couple of questions. And thank you all for joining us today.
Yeah, it doesn't look like there are any questions that have come up.
Okay.
Realized I didn't turn my camera on when I was introducing you, Sandrine. Do that now. Guess we saw as Omobola is pulling up her presentation. Guess if we have any other burning questions for Sandrine, please feel free to submit them now. I'm going to introduce Omobola Mudasiro. She is a science advisor with the American Petroleum Institute. At API, she focuses on health and exposure issues related to the National Ambient Air Quality Standards, specifically how the research and scientific data is applied in public policy decision making. Prior to joining API, she worked as an environmental health consultant for close to a decade. Welcome, Omobola.
Okay, I need to unmute myself for people to hear me, right? Well, okay, welcome everyone. I hope you can see my slides. That's a yes. All right, I'll start. My name is Omobola Mudasiro. I work with the American Petroleum Institute and I staff its health research group. We focus primarily on health and exposure impacts associated with the National Ambient Air Quality Standards, NAAQS, to support regulation that is evidence-based, of course. For folks not familiar with NAAQS, it was last amended as part of the Clean Air Act in 1990, and it requires EPA to set NAAQS standards for principal 6 pollutants. They are also known as criteria pollutants. They include nitrogen oxides, sulfur oxides, lead, carbon monoxide, particulate matter, and ozone. As you know, Sandrine has done an introduction in terms of how epidemiology is obviously critical in the policy decision-making process. And in the NAAQS process especially, we rely heavily on epidemiology, and it's actually one of the areas that EPI has been used. At API, we're continually looking and interested in opportunities to strengthen and pretty much enrich the current process. Some of these areas might include study design, transparency, as Sandrine mentioned, exposure assessment, of course, but we realize that the way EPI is used even in the NAAQS process, as heavily as they rely on it, doesn't necessarily include or discern all the information necessary for risk assessment, which ultimately impacts our ability to draw causal inferences from the published body of literature. I am not an epidemiologist, so I have to put that disclaimer there, but I do have to say I'm a user of epidemiology and I'm an interested party in its application. So although we may not always agree on the outcome, we can agree that a focus or additional focus on methodology will definitely strengthen the field. And there are rooms for improvements in terms of how we translate epidemiology into risk assessment and ultimately policy decision making. So that brings me to the Matrix workshop. This Matrix workshop was sponsored by API, and in our efforts of trying to understand exactly how do we bridge the gap between these two fields, API sponsored the Matrix workshop, which was mostly medium agnostic, and the idea behind the workshop was a focus on transparency, quality outcomes, concordance, and pretty much an instrument for environmental epidemiology. API was mostly hands-off, but it brought together a diverse group of professionals, including those in academia, those in industry, and government, of course, to try to understand how do we have an outcome that would be an impactful instrument and will empower regulatory agencies to use only the best studies for policy decision making. These are some of the folks we had in the room. I won't go through all of that, but this was just to show you that it was a room that had a diverse group of people and interested parties from different areas that were coming together to better understand how to translate epidemiology better in risk assessment. A bit of background in risk assessment. I used this slide to just highlight some of the key areas that are truly needed when we're trying to make public health decisions. You're looking at fate and transport, you're looking at exposure measurements, of course, epidemiology studies, like I said, toxicology data, in vitro, in vivo, exposure models, the whole works. But the Matrix workshop focused on certain key areas, which I'll go into further. But as I mentioned, we often use a large number of epidemiology studies with many positive attributes. A few of them would be the target species is directly relevant. There are no need for extrapolations, no need for animal models, because we have humans, humans as a case study. And then we minimize the use of animals in chemical testing. So, but in the NAAQS process, like I said, EPI is heavily relied on, but we do know that there is room for improvement. And why is there a disconnect in terms of how we use epidemiology? Why is there a disconnect between epidemiologists and risk assessors? What can we do to improve that communication and breach that communication gap? One of the things the workshop highlighted was that there was training and expertise barriers, there was technical language barriers, and both sides are often incentivized differently. Epidemiologists, as much as their work is going to be used in risk assessment or in policy decision making, that isn't necessarily the goal of the studies. It's to publish more, it's to be more recognized, it's to be more visible. And for risk assessors, as I've mentioned earlier, you can see all the different components that go into risk assessment, which isn't limited to epidemiology. So the matrix then was born out of this workshop and out of this communication as a communication tool to help translation of epidemiology. And one thing it wasn't trying to find another checklist that folks would have to go through in terms of when they were putting their studies together, or when peer reviewers are reviewing studies. I hope everyone can hear me.
Omobola, I'm sorry to interrupt. Could you maybe turn your camera off? Because I don't think your internet can keep up with both the camera and the audio. Sorry.
All right. Oh, no worries at all. Okay, so one of the things, can everyone still hear me? I hope so. So one of the things the matrix was intended to do was to serve as a communication tool to advance the understanding of risk assessment, the translation of epidemiological data, but it wasn't supposed to sort of supplant current best practices, and it was intended to be more forward-looking. Let me just make sure. All right, so this is the matrix that was born out of the workshop that I mentioned, and it had 9 asks. Although it seems very simple, it's very complex in terms of how you look at study design, how you look at reviews, how you understand the quality and how of studies that are being conducted, and how you weigh the data. So for hazard identification, for instance, let me just take that box as an example, is when you're looking at a study or when you're planning a study design, can you confirm the outcome? How is the outcome defined? What are the biomarkers of effects that are being looked at or will be looked at? And how can this be reported in a very transparent and clear manner? To these 3 core areas, the hazard identification, the dose response, and the exposure assessments, they align nicely with 3 core areas in risk assessment. As you can imagine, it's easy to throw in everything in the kitchen sink to say we want to look at how to better bridge the gap, but these were the 3 core areas that was discussed as will be most impactful. If I should take dose response again as another example, is what is the shape of the dose response curve when you're looking at an epidemiology study? How was this defined? Was linearity assumed, or were steps taken in place to ensure that the actual shape of the curve was thought about? Excuse me. You know, how do we harmonize exposure categories? Looking at the second box in the middle, was this done arbitrarily, or were there concordance across studies? And how do we describe the said direction or the magnitude of error? How was this addressed in the study, and was it taken or looked upon critically? Out of that assessment, or the matrix as I just discussed, 2 publications were born. We have a matrix for bridging the epidemiology gap, which sort of goes through what was done in the workshop and how they, I guess, reached some of the decisions that were made. And then, of course, they did NO2 as a case study of the matrix, just to see how looking at some of these studies could be improved better in terms of how we translate epidemiology NO2 studies. So the key takeaways from the workshop was risk assessors want to use epidemiological data. Epidemiologists want to have an impact, but don't necessarily know what is needed. So this matrix, as I said, was developed to begin the dialogue as small changes make big impacts. How do we improve the data? How do we improve, you know, get better asks for design, for data analysis, and for reporting? So the path forward, at least with API, is to continuously engage researchers to ensure that they consider the matrix and its elements when they're putting out their studies. As Sandrine mentioned, our journal editors looking at this, our funding agencies looking at this, and then within API to put our money where our mouth is, to ensure that when we are putting out projects, we're ensuring that the matrix is also being applied. I have a link in the to the matrix publication for folks who might be interested. And if you have any questions, please let me know.
Thanks, Omobola.
Okay.
Don't see any questions now, but as we're maybe switching to Dana, we can pause to see if there are any last minute questions. There was a question submitted to Sandrine. I think we'll come around to it at the end.
Okay, thank you.
Okay.
So I am pleased to present Dana Sargent, who is the VP of Global Regulatory Affairs, Seeds and Trays for Bayer AG Crop Science Division. She has more than 25 years of regulatory toxicology and risk assessment experience. She sent a much longer bio, but I'm cutting it down significantly. But in addition to her years of experience, she has led numerous industry technical groups investigating the science of population sensitivity, human exposure, and risk assessment methodology. She also served 20 years in the U.S. Army National Guard, where she held numerous leadership positions, including 2 unit commands. She currently lives in St. Louis, Missouri with her husband and has 2 children attending University of North Carolina. Thanks, Dana.
Good. Thank you for the opportunity to present today. I am also going to just put my camera down in case it stretches the bandwidth on the internet. So I am going to very, very briefly and at a high level just give some insight into some crop life activities that have been going on from the industry perspective around epidemiology. And in trying to move the slide. Are my slides moving or no?
Try to click on the slides with your mouse, and then you should be able to use your arrow keys.
Okay. Yep, that worked. Thank you. Okay. So I think it has already been discussed how important epidemiology can be in providing some unique information to the assessment of environmental exposures to crop protection products. But as was already mentioned by some of the speakers, there are some areas that make the integration of that information into risk assessment somewhat difficult for regulators. And so what we're doing in Crop Life, we have 3 projects that are currently ongoing where we're trying to focus on some of those areas on how exposure is assessed, how reliable some of the exposure methodologies are, and also if we can walk through an objective weight of evidence that also leverages both the epidemiological information as well as all of the animal data, in vitro, in silico information that is developed around crop protection products during the development and registration process. So I will so this slide just basically is how important it is, and also through a variety of workshops that Crop Life has had with researchers, with epidemiologists, with risk assessors. We really ended up focusing on these 2 areas, which was exposure characterization and weight of evidence. So the first project I will talk about in very, very high level, and I've given you the web link to the presentation, and I'll also give you references to some of the publications that have come out of the project, is a project that is being run by the Institute of Occupational Medicine in the UK. It's focusing primarily on improving exposure, analyzing exposure assessment methodologies, and looking for ways to improve the methods that are used in epidemiology studies. This is a multinational research project. As you saw from the beginning, the University of Manchester, Utrecht University, and also Health Safety Laboratories UK are all involved in this project. What they are doing is using existing cohorts that these institutes have been involved in, These are occupational epidemiology studies, but they're using these studies and revisiting studies that were started as far back as 1993 all the way through 2017, and looking at the exposure methods that were applied and reapplying or re-interviewing participants, as well as using biological monitoring information to test the reliability of some of the exposure methods that were used. So I'm going to just skip through because it's a rather complex project. It has 4 main work projects. I realize this slide may be way too busy, but again, you can visit the site and also take a look at some of the publications. One of the first publications was actually the protocols that are being used for this project. So the work packages, very quickly. The first work package was really to look at the exposure assessment methods that were employed and to understand exactly how those methods are used and the types of bias that comes from those methods. So it just set a foundational review of existing exposure assessment methods used in occupational epidemiology. The next 3 work packages are really the analysis and eventually looking at how they may recommend improvements to the exposure assessment methods selected for epidemiology studies. So the first work package was really to look at reliability of recall. So the original data was questionnaires and job history. And what they basically did was send out new questionnaires and really look to compare the reliability of recall. The third and fourth work packages are they look at these 4 cohorts as mentioned down below and went back to the participants and did some biological monitoring along with the questionnaires and job history to really look at the reliability and see about the validity of the different methods. And then in the fourth work package, all of these will result in multiple publications. They apply different algorithms and different ways of analyzing the information to see how they could maybe propose some improvements to the exposure assessments that were applied. Here's the publications to date. So there's been 3. The first one is that work package 1, the systematic review of the method. And then the other 2 is the overall protocol and the methodologies that they're using. And then one that is just an idea around improving the exposure assessment methodologies being applied and an overview of, excuse me, of the different methodologies that are most commonly used. So we anticipate that the further publications will come in by the end of this year. We hope to have the rest of the analysis done for work packages 2, 3, and 4, and we can provide some updates as those publications come out. Okay. The next effort and 2 projects are being run by CropLife Epidemiology Work Group, and it's really a collaboration across CropLife International, CropLife America, and CropLife Europe, because we all had various work groups looking at epidemiology and trying to think of ways to improve the use of epidemiology and plant protection risk assessment. And so we joined the resources, the available resources. We have a number of epidemiologists, toxicologists, And what we will ultimately like to do is maybe have some workshops where we invite researchers as well as some regulators to work through some of the ideas that we come up with around enhancing exposure characterization and weight of evidence. I will just quickly, because time is going fast. So the first focus area will be looking at new assessment methods for exposure characterization, where we're really trying to say, are there new methodologies out there that could enhance the exposure characterization and enable, whether it's biomonitoring or questionnaires, is there something that could be used in addition that would supplement and enable a better estimate of historical perspective exposures, particularly with crop protection chemicals? And right now we're in the process of completing out the project overview and doing a review of methodologies that could be potentially applied to exposure characterization. And we look to hopefully do that analysis, build off of what the IMPRESS project was already providing, and maybe have a publication by the end of the year. The other one was focusing on weight of evidence. And this is really to say there have been, and I know it was mentioned earlier, there have been quite a number of frameworks and guidances around approaches to weight of evidence, which would enable risk assessors to look at epidemiology along with animal data, mechanism of action information, species information, to better incorporate all the available knowledge to do an appropriate weight of evidence. And what we would like to do is take a lot of those frameworks, including ones from pharmaceuticals as well as from chemicals, and look to see if we could apply through case studies, work through a weight of evidence, focusing on the available information, the biological plausibility, and kind of identify what works well, what doesn't work well. And we're open to interested parties who may want to look at what we're trying to do and provide insights. So that's where industry is at the moment. If there's any questions, happy to take them. Okay. Are you on mute? I can't hear you.
Carol, thank you for the fantastic presentation. I don't see any questions from the audience at this moment, but I was wondering whether there's anything in the course of IMPRESS project and related work that surprised you, something that was new and unexpected.
I think we're too early yet in the analysis to see what might be different from what we had originally perceived. So we're waiting, especially with the IMPRESS project. I think the results of the analysis that will go on in work package 3 and 4, where we really look at the reliability and validity versus like biomonitoring or other ways of looking at exposure. So we're still a little early, but I'm hoping maybe by the end of this year that we would have some good insights.
Thank you.
Yeah.
Yeah, I don't see any questions again, but I'll go ahead and kind of ask you one. Is there anything more you can say? I mean, in terms of the biomonitoring samples, I assume that they're urine, but how many samples you took and how you decided to take those number of samples? Is there anything more information you can give about that?
So all of the nitty-gritty details, David, are in the protocol that they provided, but they essentially went out and collected at least 2 samples per participant that was available, and in particular, the study that was in Uganda was ongoing, so it was a good way to get in time biomonitoring samples. And they're all in the process of being analyzed by the Health and Safety Lab in the UK.
Okay, thank you.
Yep.
There was a question earlier if there are no other questions for Dana. There was a question for Sandrine.
Yes, there was a question earlier, and it was to do whether focus group addressed how to better link toxicology and EPI. And I guess I would slightly put a tilt on this question to make it more focused. Did work on the matrix or the focus groups that the committee conducted provide any new ideas on how to link the two? I mean, this is obviously been looked at as a central point of discussion. So either presenter, if could address that, that'd be fantastic. Did anything new come up in this area?
So I would say that, in these focus groups, we very much zeroed in on the epidemiology, right? So the conversation was centered around a case study that was purely epidemiology, and we poked to try and figure out what people were focusing on when they analyzed epidemiological data. For example, were they interested in seeing some exposure validation or some bias analysis, or were they interested in statistical power and effect size and this kind of thing? So we did not really sort of extrapolate on how to sort of marry toxicology and epidemiology for risk assessment. We really focused on the epidemiology, but yeah, I mean, that's definitely something that's that needs to be explored. There's no question about that.
Yes, this is Amabola. Sandrine captures that nicely. The focus for the Matrix workshop was epidemiology. I can imagine an additional webinar series or workshop could also focus on how we incorporate or how we look at toxicology and epidemiology studies for the sake of risk assessor. But for the thing I would say was interesting, I think one of the things as a user of epidemiology and as an interested party in risk assessment, was the fact that when you do ask epidemiologists what the most important thing is when they're conducting, developing a study design or, formulating studies, it's completely different, population, sample size, and all of that comes into play. But when you're comparing that to what risk assessors need, as I showed in the slide earlier, there are just so many other cocktails that goes into that. And I think it was just that disconnect that I found interesting.
Okay, thank you. There's another question here. I'm not quite sure I understand it. Maybe if the person who wrote it in might be able to explain a little bit more. The question's listed as what animals you consider in the study rate, MIC on that MIC. So if the questioner can maybe provide a little bit more details on that, but probably in the meantime, a kind of question for Dana Sargent. You indicated that you expect a couple publications out this year, 2021. I guess, have you had more discussions about what happens in 2022 and beyond in terms of the IMPRESS project and things you might be looking at, for example?
I think what we would hope for is that once the analysis and the publications are available, is that we could have some forums where we could have some dialogue, right? So because I think the ultimate outcome they would like from the IMPRESS is that they can make some recommendations for ways to improve exposure characterization, and the researchers at the different universities have a desire to continue to engage with the epidemiologists performing studies. Yeah.
Great. Okay, thank you.
And David, I would just add that, you know, if there's some follow-up research that gets identified, you know, we would probably reevaluate that under CropLife Europe as far as continued support for, you know, additional investigations and efforts.
Okay, great. Okay, thank you. I don't see any more questions, but I'll go ahead and ask one, and that is, so you focused on two things, kind of the reliability of the exposure measurements and also the weight of the evidence. Were there other things that, can you say anything about why you focused on those two things, or were there other things basically that you kind of talked about that that might be of interest, basically, that maybe you left for later, or you didn't think it was, you didn't do or couldn't do on that? So maybe just some thoughts back from you about how you decided on these two and what you may not have looked at.
Yeah, so I think, you know, we did spend a fair amount of time looking at the most current guidance. There's been some really good review articles on statistical methods applied, evaluating bias, study design, good practices, and things like that, all of which we felt were informative and in a direction that would continue to improve our ability to use information out of epidemiology. And so we didn't want to reiterate on work that we felt was already providing some really good insights and direction. So what we found out of the available guidance, though, and frameworks, was there wasn't maybe a really clear look at, is there new methodologies, new types of methods that could be applied that would be economical, easy, reliable, and would supplement the exposure characterization and maybe help in making it a little more quantitative rather than qualitative. So we wanted to see if we couldn't investigate that. And then with the case studies, there's some frameworks out there, and what we wanted to do was, being industry and the folks who own a lot of the data that is used to develop these compounds, looking at mechanism of action, looking at dose responses, knowing a lot of the detail, and also even looking at medical information on medical use of plant protection products. We wanted to see if we couldn't walk through some really good case studies that then could further the conversation around how do you go about integrating epidemiology for human health risk assessment. That's why we focused on it. We just felt like these were areas we could continue to move forward, and other areas, I think there's been a lot out there already.
Great. Okay, thanks. And by medical uses, you mean things like the conazoles and the sulfonylureas, for example?
Exactly. Yeah, okay. And even the pyrethroids, or yeah.
Yep.
There's another question here.
Yeah, there's another question, and I think it's addressed to, I think, all the presenters, and that is how, you know, have groups discussed improvements in better documentation of hypotheses, as well as validity of models for exposure risk relationships that are being presented in publications. So two really rather distinct questions, how hypotheses are captured and better way to do so, and whether valid exposure response or exposure risk models are being applied. So any thoughts on those two big topics would be greatly appreciated.
So I mean, I can start, but this is not really something that we discussed. Our conversations were, I would say, at a higher, broader level. We did discuss exposure evaluation quite a bit, and talk about the trade-off between having a robust exposure evaluation versus trading that off against study power, sometimes, and the fact that often having a robust evaluation may not help having a study funded, for example. But this question is a bit more specific, and that's not something that we address specifically.
So I might call out on Raga as far as exposure models, maybe. I mean, we clearly look at how exposure information is captured and how reliable it can be in actually estimating exposures, real exposures. And certainly we've found it difficult to take the current methodologies and turn them into quantitative estimates.
Yeah, I can try to answer that. Yeah. So part of the CLI, CLE collaboration, one of the work streams is looking at NAMs for exposure assessment in environmental epi studies for crop protection products. And yeah, we're kind of still evaluating different methods that have been used for exposure characterization so far. And yeah, it's not straightforward because these are not animal models where what you're giving or dosing the animal, what dose you're giving. So there's always going to be indirect methods and questionnaires and matrices. So like Dana said, we're also looking at outside environmental epidemiology, like pharmacology, pharmaceutical industry, to see if we can use different exposure metrics and try to compare with what we have. So it's still developing. I hope that answered the question.
And if I can jump in, I hope everyone can hear me. I was saying that here at API, we haven't necessarily focused on hypothesis generation, but we are definitely looking at how to better look at methods that look at exposure characterization. We have a few RFPs going out that we're going to be putting out in about a month or so, where we're hoping that we can get folks who can provide additional information on how to better characterize time activity patterns, models that include indoor air characterization, particularly given the times and the amount of times people are spending indoors now, given the pandemic and all. But how do we better capture and how do we improve the existing data in this space? And then, of course, translating or looking at how it impacts risk assessment forward. But yes, it's definitely important, at least for us.
And you expect to do that probably with surveys? Is that the kind of information you'd be gathering that information from, the time activity patterns and such? Or anything electronic, mobile phones or anything like that?
Exactly. You're absolutely right. We're keeping the RFPs very open because we don't want it to be prescriptive, but we're definitely looking at our proposals that incorporate technology and how that can be used to capture some of these activities or exposures, so to speak.
I don't see another question, but I'll go ahead and ask one for Dana. But anybody, I guess, that might want to answer. One of the things that a lot of the epi studies we look at, basically, they'll do a continuous measurement, for example, sometimes, but they'll break it, basically dichotomize it, or break it into tertiles or quartiles, for example. Do you have any thought about, basically, I mean, if you're doing the biomonitoring, you'll have the continuous measure, doing some kind of comparison about how the results may differ, say, between taking breaking things out into categories versus, for example, continuous measures on that, and doing epi with those? Have there been any discussions or thoughts about doing those kinds of comparisons and looking at the potential differences?
Oh boy, David, you're challenging me.
Yeah, I can try to answer it. I mean, yeah, that's a very good idea. We haven't talked about that within our subgroup yet, but that's something that we can think about, potentially collaborate with the EPA with, if you're interested in. We can initiate a discussion on that within the group when we have the next call, and then, you know, maybe we can start thinking and going in that direction as well.
Okay, thanks.
Mm-hmm.
All right. Igor again here. I don't see any questions, but I will ask one of my own, if I may, and that is, again, you know, I would welcome all three presenters to comment on it, and whether you think the difficulties in connecting epidemiologists to risk assessors come from the way in which epidemiologists collect the data, or the way in which they treat and present the data. So what is your sense? If we're to make a rapid improvement, would it be in the collection of information, or in the way that information that has already been gathered is treated and presented? Thank you.
That's a very good question, Igor. I think if I was to pick one, I would say it's the way the data is being collected, because I feel like where you can have the biggest impact is obviously at the very front end of your process, where you can ask the right questions, define the categories properly or accurately to answer specific research questions. So if we, and that's why we really like what we did with the matrix, is that if you can have epidemiologists think about these criteria, or I guess ideas, so to speak, at the front end, it would make the studies more impactful or more applicable or useful and more translatable in risk assessment. But I would say the bulk of the work and the bulk of the efforts will have to come in the beginning, the collecting data and the study design. And yeah, that's what I would say if I was to pick one or the other.
Yes, and I would agree. I mean, I want to say both, but I think that's why it started, the data collection. And I think what became obvious after these focus groups is that epidemiologists do risk assessment is not on their radar. So they are not designing their studies with an eye towards potential data, potential application in risk assessment. And if they knew how much their data, how much of an impact their data could have in that field, I think they might collect their data in a different way. So that's where it starts. And then, of course, the way the data is reported matters a lot also. Yeah, I would agree on Omobola. I mean, if we have to start somewhere, that would be on the data collection and study design.
And I would be, you know, bold enough to say we have to recognize that in order to meet the needs of risk assessment, the data collection becomes much more onerous, right? What we need and over time, and even you know, David's question, right? when we're looking at what type of data gives us the best true estimate of exposure, and how do we take it over time and across the population. So we have to also think of ways in which we can kind of meet in the middle, which is understand what we need for risk assessment, maybe make some adjustments for uncertainty, but also make it reasonable that it can be managed in an epidemiology study with the resources that are typically available, right? Or come together and decide to do a much bigger investment to make a more robust design that would enable good, solid risk assessment.
Great. Thanks, Dina. It is 2 o'clock or 2:01 now. Unfortunately, I do see another question or two, but I think given time, I think we're going to have to wrap up here. So I'm going to thank everyone for joining and thank the 3 presenters, Sandrine, Omobola, Dana, for your presentations and your thoughtful answers to the questions and the discussions. I will just close with this last slide. Again, thank you for everyone for joining. Just a reminder that the recording of the webinar will be available in a few days on HESI's Environmental Epidemiology page. We do have other webinars planned. The next one will be scheduled for the end of May or June. If you have any questions or feedback, please contact Sandrine. So thank you, everyone.
Thank you.
Thank you very much.